Okay, okay. So I repeat again. One, this one. Okay. So if you want to have information about receptors, as I told you before, it's not possible to have the crystallized form of these proteins. We can investigate, we can perform some modifications about the chemical structures of particular ligands, for example, endogenous ligands, or other molecules that we know are able to interact with this particular receptor. It's also possible to make a recognition of the active site by small modification on the chemical structure of an agonist. It's the same situation when you enter in a dark room and then you try to know where is the window, where is the door, where is other things located on the walls. You try to see how the room is organized. It's the same. We can use a chemical structure. We can add particular groups on the backbone to explore the active site of a particular receptor. Here, you can see how it's possible to get a crystal of a specific protein. It is necessary to crystallize this protein, and so it's possible, it's very easy with enzymes, more difficult with the for example, membrane receptors. When we have the crystal structures, we can perform a diffraction investigation. You have studied during the physics course what is diffraction. The different electron density present inside the crystals are able to change the direction of specific light ray. You know how works diffraction. Yeah. Okay. Can see better. Yeah. Okay. So you see, you put your crystal under a specific equipment, and after the interaction of particular rays with your crystal, it's possible to build a map with the different atoms of a particular protein. It's very easy for enzymes because enzymes are isolated structure, isolated proteins. So we can extrapolate this type of proteins from the environment and their structure remain the same. Okay. But if we have a membrane receptor, it's very difficult to extrapolate this protein from the environment, because normally it can change its tertiary, quaternary structures, and we get very few data about its arrangement in the space of the different amino acids. It's very important to refine the data obtained by a diffraction investigation using a computational approach. In Bologna now, there is the most important center of computational investigation, of drug computational investigation in Europe. The most potent. How it works? It has a lot of protein structures. It can work by analogy, and so it's possible to build the model of particular proteins by analogy, to put together all data and to have one model. This model should be validated by virtual interaction with a specific ligand. Nevertheless, it was possible to have some models of specific receptors. The first receptor crystallized was the nicotinic receptor from Torpedo californica. Is a fish. This fish has a very big nicotinic receptor, and it was some years ago possible to crystallize this type of receptor, and so we know that nicotinic receptor is a channel ions receptor. You see here on the picture the model. Some years later, also the adenosine receptors was crystallized. This is another type of receptor. You can see in the picture, this is a 7 transmembrane receptor, metabotropic receptor. After that, beta 2 adrenergic receptor was crystallized. And in 2020, Nobel Prize to a group of scientists because they were able to carry out, to prepare a specific experiment, to have a snapshot, to have a picture of specific proteins. You see the title, Revolutionary Cryo-EM is taking over structural biology. In particular, they were able to reconstruct the 3D, three-dimensional shape of particular proteins. Here you see the difference. Resolution before 2013 and the resolution at present. Why it was possible to have this deep resolution, because the problem is related to the movement of the proteins. So if you make a snapshot of a protein at room temperature, it's not so clear the structure, because the proteins normally are not immobilized. But if we make the same picture at very low temperature, it's possible to frozen the biological structure of a protein and to have a deeper resolution, a higher resolution with a deeper information. So, starting from these new techniques, now it's possible to have more information about particular proteins. You see, Rosetta Fold is the name of this technique. Accurate protein structure prediction accessible to all. So it's possible to put this information on a website, and so all scientists can use this type of information. Once again, you can see the information that you can find now on the web. When we know the chemical structure, for example, here you see the nicotinic receptor, we can design new molecule, and we can know the site for the agonists, for example. We can use particular ligand. You see this polyamine chain. We can use particular probes, we say. Probes are molecules able to interact with specific part of a receptor. Some years ago, we worked a lot about nicotinic receptors, and in that time, we wanted to have additional information about agonist sites, and we use this type of probes. A probe is not a drug. A probe is a molecule able to interact with a specific site of the receptor, but we cannot use this type of molecule as a drug, no. These probes are able to give a covalent bond with a particular site. Normally, they are labeled with radioactive atoms. And so you can incubate your receptor with this probe. This probe is able to bind to the receptor covalently. And then you make a digestion of the protein. We investigate, just a moment. After the digestion, we have a small part of the receptor, and we can see where our probe is bound because it is radioactive. So we can have additional information about a particular interaction site, binding site. Just to give you an idea about the studies, the different investigations that now are carrying out on this topic. If it's not possible to have any information about the receptors, we can investigate, we can perform investigations about particular ligands. As I told you before, we can start from a endogenous ligand, acetylcholine. We can modify the chemical structure of acetylcholine in order to verify if these modifications can increase the activity or not. Here you have an example with an exogenous molecule. Is a flavonoid. Flavonoids are natural compounds, very important because they are antioxidant. And so they are very common in cosmetics, you know. If you want to preserve our skin from insultants, from pollutants, and so on, okay, we can use antioxidants. But some flavonoids are agonists of particular receptors. And so in this picture, you can see a classical structure, activity, relationships study. We take the backbone of a flavonoid and we perform a lot of modifications, chemical modifications on this structure. You see different substituents. For example, in this position, air, prenyl group at these positions can increase the activity. In this other position, OH group in these positions can increase the activity. Substitution of methoxyl with OH can increase the activity. OH group at this position are vital, are important for activity. So this is the picture. This picture describes a classical investigation of structure-activity relationships on a specific molecule. This is the classical task of a medicinal chemist, because in lab you have to synthesize a large number of analogs with a small difference. For example, in this position, we can put a methyl, an ethyl, normal propyl, isopropyl, and so on. So we can investigate this part of the molecule. And we have information about the environment on the binding side that we don't know. But we can get information, additional information, and sometimes we can have a more potent compound, more potent agonist, more selective, you know. So we have seen the two different approaches to have information about a specific target. Crystallized model, computational and crystallized model, okay. Very easy to design new drug because we have a picture of the binding site, and with a particular software it's possible to design specific ligands. Otherwise, if we don't have any information about the active site, we have to use the classical approach of medicinal chemistry called structure-activity relationships. Structural modifications to see what happens in the interaction. Is it clear? The two approaches, okay? Something about the history of the receptors concept. The first scientist who described for the first time this concept was Langley. You see the name? He was a student, a physiology student at Cambridge University. 1878. He had an idea. In that time, he studied the interaction of atropine and pilocarpine, you know, two important natural alkaloids, with in the cats. And in particular, he studied the influence of these two compounds on the saliva formation in the cats. And he hypothesized that these two compounds, in some ways, interact with particular substances inside our body, but no any other evidence, just an hypothesis. Later, Paul Ehrlich, another important scientist, suggested that the cells have particular side chains on the membranes able to interact with drugs. The first model of the receptor. Particular side chain on the membrane of the cells. In 1905, Langley, the same student, but now Langley is a professor, and okay, that time Langley was some years later was a professor, studied the effects of curare. You know, it's another toxin, natural toxin, able to block the movement of the muscles, and concluded that there was a receptive substances in the body that would receive the stimulus and then cause muscle contraction. So these two scientists, the contribute of these two scientists was to outline a model, the fundamental characteristics of the receptors. This means the ability of the protein to recognize specific ligands and the ability of the ligand receptor complex to initiate the biological response. This is the most important point. A receptor is a protein which can interact with particular molecule, and only after the formation of the ligand receptor complex it's possible to have an activation, to have a biological response. But what kind of forces are involved in the drug receptor interaction? Normally, non-covalent bond, normally. And normally they are weak interactions. These interactions should be reversible in order to avoid a long-term effect on a receptor. Normally, after hour, minutes, the effect should stop. Once again, we have the two definitions of what is an agonist and what is an antagonist. In this system, if a drug is able to simulate the action of a natural mediator, acetylcholine, for example, we have an agonist of the acetylcholine. If this molecule is able to block the activity of an endogenous natural mediator, we say that we have an antagonist. These are two important concepts, agonist and antagonist, because in the specific parts of the course we'll see a lot of agonists, but also a lot of antagonists of the same receptor. We have seen that the interaction of a ligand with the receptor is due by weak forces, weak bonds. But why it's possible to have selectivity, to have activation, if only weak forces are involved in this interaction? This is the question. Because it is important a strong complementarity of the target and the ligand. If we don't have a strong complementarity between these two entities, we have no interaction. You see here an example. C is the target, but also D is the main target. So the entity A, this compound A, is able to interact with C, yes, sometimes. Something with B. But the highest complementarity is with target D. So we have a complete interaction with the target D, yes, not B, D, as David. And in this case, possible to have the activation of the receptor or the block of the binding site. In this case, we have an antagonist. In the other case, we have an agonist. You see, selectivity of action can be achieved by the involvement of different interactions in the formation of the DR complex. Drug receptor complex, in this case, DA interaction. So now, I'd like to describe you the different classes of receptors, but I hope that you have already heard, that you have already seen this type of receptors, for example, during the course of biochemistry, during the course of physiology. I am a medicinal chemist, so I can give you a treatment, an idea of these receptors from my professional point of view. Two main classes, membrane receptors and intracellular receptors. Why we use this type of classification? Because membrane receptors are located on the membranes in aqueous environment. So normally it's easier to reach, to interact with this type of receptors. Intracellular receptors are inside the cytoplasm. And so if we want to reach this type of receptor, we have to cross the cell membranes. And we have already described the difficulties present which a compound has when try to cross the cell's membrane. It's not so easy. We need, you remember where the two personalities of a drug, lipophilic and hydrophilic. And so we'll see the different chemical structures of the different molecules, first class molecules able to interact with the membrane receptors, second class molecule able to interact with intracellular receptors. Here you can see different types of receptors, membrane receptors, G protein linked receptors, serotonin, muscarinic, dopaminergic, noradrenergic, they take name from the agonist, endogenous agonist, serotonin, acetylcholine, dopamine, noradrenaline. There are also present enzyme receptors, tyrosine kinase, involved normally in cancer in the growth cell, the cell growth, or otherwise in the cancer cell reproduction. Ligand-gated ion channel receptors, nicotinic, GABA, glutamate. We see some agonists or modulators of this type of receptors. Intracellular and nuclear receptors, hormone receptors, autocoid receptors, growth factor receptors, insulin receptors. During our course, we don't meet this type of receptors. You will find this type of some agonists of this type of receptors in the medicinal chemistry too, hormones and so on. Membrane receptors, ligand-gated ion channels, G protein coupled receptors, tyrosine kinases, integrins, and so on. First class, G protein coupled receptors. You know that these receptors are formed by a long protein chain normally 700, 600, 700 amino acids. And this chain is able to cross for 7 times the cell membrane. As I told you before, it's not so easy to know, to have information about the tertiary or quaternary structure of these proteins, these receptors. The model, the first model of these receptors was built in around the 80s by studying the rhodopsin receptors in eyes, inside the retina. You know there are particular receptor, light receptors. And the rhodopsin was the first model of this type of receptors. Here you can see the different examples of G protein coupled receptors. Okay, it's in Italian, but we say neurotransmitters, you know, adrenergic, dopamine, serotonin, the different G proteins coupled with this type of receptors, the effects, the final effect, phospholipase C, adenylated cyclases, potassium channel, calcium channels, and so on. We'll see during the course compounds able to activate the dopamine receptors or to block dopamine receptors, to increase the activity of the serotonin receptors, yes. And also you see eicosanoids, prostaglandins. These receptors are involved in the inflammatory process, and we'll see molecule able to block the formation of prostaglandins, which are the agonist of this type of receptors. Okay, we have now a brief movie, just a moment. I want to show, okay, now, I have to make again, just a moment, here, and then I find, so, include the audio, okay. Okay, so, now we can see together this short movie.
As a receptor site for a small molecule called a ligand exposed on the outside of the cell. The portion of the receptor on the inside of the cell can bind to the G protein, which has a guanosine diphosphate attached to its alpha subunit. When the ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. This step can be repeated as long as the ligand remains bound to the receptor. When the ligand separates from the receptor site, additional G proteins are no longer activated. Inactivation of the alpha subunit occurs when its own phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the subunit. The G protein subunits then recombine and attach to the receptor in the cell membrane.
Okay. So you have seen how this type of receptor works, how is it important to have the right agonist in order to activate the second messenger of these receptors. Now we can see the ion channel receptors. These type of receptors are faster than the metabotropic receptors. These receptors are represented by a channel which can be opened very fastly, and so we can have an inward, a strong influx of different ions, positive or negative ions. If positive, you know, we have an activation of the cells. If we have an inward of negative ions, chloride ions, for example, minus chloride ions, we have a hyperpolarization of the membrane, so we can stop the activation of the cell. One of the most important class of these receptors is represented by nicotinic receptors. Positive ions flew inside this type of channels, so we have an increased activity of the cell. Once again, we see another short movie about this type of receptor.
The sodium channel has receptor sites for a ligand acetylcholine. When the receptor sites are not occupied by acetylcholine, the sodium channel remains closed. When acetylcholine binds to its receptors, the channel opens. Therefore, the channel is called a ligand-gated ion channel. When the channel opens, sodium ions diffuse through and enter the cell. The sodium channel has receptor sites for a ligand acetylcholine.
Okay, so you have seen again how this type of receptor works, and so we can increase the content of sodium in this case by the opening of this type of channels. The last class of the receptor is represented by tyrosine kinase receptors. These are very fast receptors involved mainly in cancer cells replication. So there are particular drugs able to block this type of receptors in order to decrease the speed of replication of cancer cells. Okay, we are at the end of this chapter. Have you any questions? Comments at home? Nothing? Okay. So, just a moment. Okay. All clear? Off. Okay. Okay, Stephanie. Okay, now we start with the second class of receptors. Intracellular receptors. As I told you before, these receptors are located inside the cytoplasm and or inside the nucleus. So normally, for a compound, it's very difficult to reach and to interact with them, because this compound should cross a lot of different membranes. External membrane, nucleus membrane. And so normally this type of molecules are very lipophilic. Here you can see an example of this type of receptors called retinoid receptors. Retinoid receptors are inside the nucleus, very close to the DNA, and after their activation, we have an increased activity of synthesis of new proteins. Some natural compounds are able to activate this type of receptors, and their activation can lead to an increased speed of cell reproduction. These results can be useful in anti-aging compound in cosmetics. If we want to repair our skin because it's old, we want to restore a young appearance of the skin, we can increase the replication of particular skin cells. This is an example of retinoids. Otherwise, some anti-cancer compounds are able to interact with these receptors to control or to increase the biosynthesis of particular checkpoint of the cells. This is a very important point. However, retinoid receptors are inside the nucleus, so it's very difficult to reach them. The most common receptor of this type are hormone receptors. So you see a lipophilic hormone normally interact with the cytoplasmic receptors, and this complex can reach the nucleus, can interact with the DNA to give particular results, normally an increased speed of transcription, and so an increased number of synthesized proteins. Once again, a movie about these receptors. It works, I don't know. Perhaps no. We try again. Doesn't work. Okay. Okay, sorry. However, you see the DNA, nucleus, cytoplasm, external membrane. You see the hormones molecules here. Hormone molecules are able to cross the external membrane to reach the cytoplasm, to interact with specific receptors, and to go directly to the nucleus to give their action directly on the DNA. And we have an increased synthesis of messenger RNA to have an increased number of synthesized proteins. So now The next topic is related to drug receptor bonds, but I think it's better to stop for 15 minutes. So if you don't have any question at home or here in classroom, have you any questions? No? Okay. So we can start together 11:30, okay? So we can take a coffee. And then we'll see the different receptor bonds. Okay. So see you later. Then we stop for the moment. So, dear students, we start again. We have seen the different drug targets. We have seen how these targets are formed, are organized, where they are. And now we discuss about the interaction, the forces involved in the drug receptor interactions. First of all, I'd like to remember that there are some drugs active at very, very low concentration. You see, this low concentration, picomolar, some toxins, botulinum toxin, they are active at very low concentration. This means that these compounds are able to interact with the target, yes, to activate these targets, yes, in a very potent fashion. So we have to know the different forces involved in the drug receptor interaction. But the first concept is that the chemical structures of the two subjects, drugs and receptors, should be complementary. You see here in the picture how it's important the complementary of the two chemical structures, and when a drug is very close to the target, it's possible to have a mutual interaction, and so perhaps also the target can have small modification in order to have an optimal interaction with the drug, with the molecule, with the agonist. This is due by the different forces involved in interaction, the influence of these forces on the drug receptor interaction. There is an important concept: no action without interaction. This concept was introduced by Paul Ehrlich a lot of years ago, several years ago, you see, 1914. And at that time, the scientific papers were written in Latin. In Latin we say, corpora non agunt nisi fixata. This means no action without interaction. So it is necessary an interaction of the drug, of the agonist, of the antagonist with the target. Normally, the forces involved in this interaction are very weak, but many weak interactions make a strong interaction. Two years ago, there was a national strike in Italy. If only one person is on strike, nobody knows your problem, your situation. If an entire nation is on strike, perhaps the government try to decide something to solve the solution, to find a solution. So it's the same in medicinal chemistry. Many weak interactions make a strong interaction. Normally, the forces involved are weak, okay, weak bonds, easily reversible, but after the interaction, normally the complex evolves towards more stable and not towards irreversible bonds. What kind of bonds are involved in the drug receptor interaction? DR means drug receptor interaction. Covalent, not so common. Electrostatic, ionic, very common. Hydrogen bond, very common. Perhaps you already know this type of bonds. Charge transfer, perhaps for you is new. Hydrophobic, for you is new. But you already know van der Waals, weak bonds, but a lot. Dipole-dipole, ion-dipole interactions. Now we'll see all these types of bonds and with example.
Yeah.
Yes?
But we said that it should be a weak interaction.
Yeah.
But not actually a strong.
Okay. Okay. Normally, weak bonds are involved. Van der Waals, for example. Van der Waals is very weak, you know.
Yes.
But a lot of van der Waals interactions make a strong interaction, because you have to consider the surface of the drug, and so there is not only one, but several. You put all together, the final result is a strong interaction. Okay? Not only one. Van der Waals bonds, you know, they originate from transient dipoles. The electronic clouds can have different density, electron density, and so you can have a positive part of the cloud and a negative part of another cloud, and so they interact, the positive part of a cloud with a negative part of another cloud. You know? Very weak, yes. They decrease exponentially with the distance. So it is important that the molecule, the distance of the molecules from the target. This type of bond is important in drugs with aliphatic chains, aromatic groups. This bond is involved also in the stabilization of protein molecules, tertiary structure, stabilization of cell membranes. The long chain of phospholipids interact with this type of of bonds. Ionic bonds, strong. But why ionic bonds? Because a lot of drugs have ionizable group, acidic group, CO minus, or basic group, positive nitrogen, protonated nitrogen atom. Also, you can, you have, you know the chemical structure of acetylcholine. Acetylcholine is has a quaternary nitrogen atom, has a positive charge, and this positive charge normally interact with a negative charge in the binding site. So there is a acidic residue of aspartate, glutamate inside the binding site. So it's possible to have this type of interaction, anionic bond. You have studied this type of bond during the course of chemistry, but in medicinal chemistry, this bond is not so strong. Why? Because we are in an aqueous environment. You know the solvatation. Water is able to be around a positive charge, and so perhaps the interaction between the drug and the target is not so strong in this environment. And also we see there are steric effects, because sometimes the charge of the target is located inside a gorge, a tunnel, so perhaps it's difficult to reach this site for steric effects. We see now some examples. Here you can see the steric effects. The minus charge can be located, you see, at the end of a tunnel, of a gorge. And so if your drug has a positive charge, and if your drug is just a little big, it has a big shape, it's difficult to have an optimal interaction. Here you can see a classical example of a positive charge, a nitrogen, a protonated nitrogen atom, interact with a carboxylic residue. And in addition, you see there is also a hydrogen bond. So in this case, we have two different bonds together, an ionic bond together with an hydrogen bond, reinforced by hydrogen bond. Here in the picture, you see an example of this agent called p-vagabina. Okay. In this case, you see that this drug has a acidic function with a minus charge, able to interact with a residue of arginine, is a guanidine group. It is peculiar of arginine. In this case, the group is very basic, 3 nitrogen atoms. It is protonated, a physiological pH, you see the positive charge, and so in this case, you have an ionic bond formation between the two species. the chemical formula of acetylcholine, positive head, quaternary nitrogen atom, permanent positive charge, ionic bond, but also with the tail, you see with the ester group, it's possible to have other weak interactions, London, van der Waals, and so on. Here you can see a lot of ionizable group located on the surface of the different proteins. And so these groups are responsible of the formation of ionic bond. You see the pKa, you see the percentage of ion at the physiological pH. So a lot of groups are able to give this type of bond. Dipolar bonds, dipole-dipole. You know that when two different atoms are bound together, you have a difference in electronegativity. Here you have an example with a ketone, the carbonyl. Carbonyl, you know, oxygen is more electronegative than carbon atom, and so you have a different distribution of the electronic cloud. In this case, you have the oxygen has a partial negative charge, the carbon a partial positive charge. This means that this ketone is able to interact with another ketone where the negative oxygen interacts with the positive carbon atom, and also the second oxygen is able to interact with the second carbon atom. So we have two different dipoles able to interact together. There is also another type of this bond called the ion dipole. Ion, because if you have a quaternary permanent positive nitrogen atom, this positive entity can interact with a partial negative oxygen of for example, of a ketone. Induced ion dipole. If you have, once again, a positive permanent nitrogen atom because it is a quaternary substituted nitrogen atom, this can influence another nitrogen atom very close, and so you see the electrons can be shared by the two entities, and so it's possible to have this type of bond. Here you can see an example of this drug called zaleplon. Here if you have an example of dipole-dipole between an alcoholic group, OH, and nitrate group, here you see C triple bond with nitrogen. This bond is polarized. Nitrogen is negative, partial negative charge, and carbon positive charge, and so the positive carbon can interact with the oxygen, partial negative oxygen of alcohol function. Here you can see also an example of a ionic dipole interaction. You have a residue of a carboxylic acid with a minus charge. This minus charge can interact with this carbon positive, partial positive carbon atom of the ketone. No, it's an amide. Okay, with the amide. Here you can see a table with the different dipole of the different bonds. So, for example, the strongest, where is this 3.9 sulfur with oxygen, 3.5 carbon with nitrogen, 2.6 carbon with sulfur, and so on. So we can have an idea about the entity of this bond, depending on different atoms involved in this type of bond. Hydrogen bond, you know, hydrogen bond is responsible of the boiling point of water. It's very common. But this bond is very common in drug receptor interaction, yes. Several atoms of biological interest are involved, yes. Here you have an example of amine. Hydrogen can be shared with an oxygen atom, ether in this case. This bond should be the two elements, the two compounds, the two atoms, sorry, involved in this bond should be on the same plane. If the two atoms are not in the same plane, this bond can be weaker. And so it's important. Here you have a series of different examples of hydrogen bonds. You see a residue of a tyrosine can interact with a residue of carboxylic acid, cyanized carboxylic acid residue. Two alcoholic groups can interact together, yes. Once again, you have an amine with a carboxylic acid, you will have ionic bonds, but also in addition there is also an hydrogen bond. Here you can see an alcohol with an oxygen of a carbonyl. Here an oxygen with a proton of an amide. This bond is very important to stabilize the tertiary structure of proteins. You know the different shape of the proteins, alpha helix, beta sheet, you know, and this type of bonds is able to stabilize the different shapes of the protein. So it's important. Once again, I want to outline the importance of this bond, but also the environment around this bond, aqueous environment. So the molecules of water normally are all around of the atoms, of the two atoms, and so sometimes this type of bond is weaker, can be weaker. And also, if you remember, this bond is very important in the DNA because the different bases are bound with this type of bond. This bond can be present inside the same molecule. In this case, we say that this bond is intramolecular. You have here an example with salicylic acid. You see the OH is able to interact with the oxygen of the carbonyl, the carboxylic group. But also this bond can be intermolecular between the OH, or the acidic group, with the alcoholic OH. Sometimes the intramolecular hydrogen bond is responsible of the low solubility of particular drugs. We'll see a specific family of antibiotics called the tetracyclines. Tetracyclines have a lot of OH group on their structure, but the hydrogen bonds are intramolecular, and so the result is that these alcohols molecules have a very low solubility in water, because all the hydrogen bonds are inside the molecule, intramolecular bonds and not intermolecular. Energy of hydrogen bonds, you see a different energy. Charge transfer bonds. This perhaps for you is new. When a drug is very close to the target, it's possible to have a migration of the electron cloud from the drug to the target, or vice versa. We have a transfer, a migration of electrons from a donor to an acceptor. Perhaps you can better understand this bond by to see these examples. You see that there are a lot of compounds rich in electrons, for example, pi electrons, unsaturated compounds, aromatics, heteroaromatics. We can have aromatic rings with a lot of substituents, with the inductive and mesomeric effects. And so we can have a high density of electron on this type of ring. And this cloud can migrate to the target, where perhaps we have a deficient aromatic systems, because sometimes there are some substituents which are able to take part of the electrons from the aromatic rings. In this way, we have two aromatic rings. The first one is very rich in electrons, the second one is very poor, and so we can have an optimal electrostatic interaction by these two rings. So we can distinguish donors, rich in electrons, and acceptors, poor in electrons. You know that in the DNA there are a lot of bases rich in electrons. So it is possible to have molecules able to interact with these electron clouds because they are poor in electrons. So this is an example. Chloroquine is an anti-malaric agent, is able to interact with the DNA bases by this bond. there is another important bond able to regulate, to modulate the interaction of a drug with the target. It's called hydrophobic bond. This bond is not a classical bond. No ionic, no covalent, no. It's quite different. It is regulated thermodynamically. It depends from the free energy variation. You know the Gibbs rule. You know that we need a decrease of free energy if we want to have a spontaneous reaction. Here you can see the expression. The variation of free energy depends on the variation of enthalpy minus the variation of entropy. So if we have a strong variation of entropy, we can have a spontaneous reaction. When I think about entropy, I think about my daughter in her bedroom Saturday evening. She tried all clothes, all shoes, everything, and everything remained on the bed. So in this case, there is a strong increase of entropy in this room. And this strong increase of entropy requests strong energy from my wife to stabilize at the end, to put everything in the right place. Okay. In this case, the same. Why? If you see, we have two molecules with different color, and each molecule has around a lot of water molecules, ordered. You see, ordered water molecules, a lot. If these two molecules interact each other, the number of water molecules is decreased. So we have less ordered water molecules. We have a decrease in the number of ordered water molecules when the two big molecules interact each other. So in this case, we can say that we have an increase in entropy. This means that we have a decrease in free energy. So this reaction is spontaneous. When you put oil in water and you try, you mix and you wait, you can see the formation of the two layers again, because you know that oil and water are not mixable. And this process is regulated by this type of bond called hydrophobic bond. Hydrophobic means that two hydrophobic species lipophilic interact each other in an aqueous environment. This means that if you have a lipophilic drug with a lot of aromatic rings, okay, and these drugs try to interact with a cluster of aromatic rings present on the surface of a protein, because we are in an aqueous environment. So I can understand that this bound is regulated by van der Waals, London forces, and so on, but we consider the total effect of these weak forces, and the total effect, the total result is that we have an interaction of a lipophilic drug with a lipophilic cluster present on the receptor, the binding site, yes. So when we describe the interaction of a drug with a target, normally we say that there is this hydrophobic bond, the formation of this bond, instead of to describe van der Waals, London interaction, and so on. Dipole, dipole. This is a summary. This bond comprehends all these weak bonds. You see here in the picture the aromatic residue of located on two different parts of a proteins. They can stabilize a particular conformation of the proteins by this hydrophobic bond. Also, you know that a lot of amino acids have lipophilic chain which can interact with hydrophobic bonds with particular ligands. Is it clear, this bond? Yeah, at home? Is okay?
Yes.
Yes, okay. Because it's a new concept for you, and so I want to know your opinion. Okay. Covalent bond, you already know this type is the strongest bond, chemical bond. It's very difficult to break this type of bond because it requires a high energy. But normally, very few drugs act by this bond. Yes, there are some examples, but it's not so common. High energy, okay. Here you have an example of particular compounds called the nitrogen mustard, able to interact with the DNA bases. These compounds are able to give a covalent bond with guanine, and this class of compounds is able to block the two helices of DNA by forming two covalent bonds with the two helices. In this way, the DNA polymerase is not more able to read the two helices because there is this molecule, this mustard, which block the opening of the double helices to be read by the DNA polymerase.
Prof?
See? Yes?
Are these anti-cancer drugs?
Yeah, yes, we'll see, yes. We'll see this type of compounds in the next chapter, in the chemotherapy chapter, as anti-cancer agents, yes. They block the DNA helices of cancer cells, so they go to apoptosis. You know what is apoptosis? It's a programmed death. And so we can kill the cancer cell by using this type of compounds. But they are very toxic because they can also interact, they can also hit healthy cells, and so we can have severe side effects. We'll see very briefly the mechanism, but because we will see in the details this mechanism in the next part of the course. You see that this compound, you see the chemical structure, very simple. It's like an amino acid. You see here, this is the residues of phenylalanine, for example. You have this nitrogen atom with two carbon atoms and an halogen here. The nitrogen, basic nitrogen, is able to give an attack to the last carbon atom because it is activated by the presence of the halogen. And we have this tensionated ring. This ring, very reactive, this electrophilic ring, you see the positive charge, can be attacked by the positive nitrogen of guanine. And you see the formation of a covalent bond. And then we have a second group here that it is able to give a second azeridinium ring, is called this ring azeridinium. And then we have a second attack from a second guanine inside the DNA. Okay, now we are not interested on this mechanism. It's just to give you an idea about the potency of particular compound able to give this potent bond in the binding site or on the DNA basis. Chelation bond. Perhaps you already know a lot of compounds able to give particular chelates with ions, metal ions. You see here penicillamine, penicillamine. You see these two groups, the amino group and the thiol group, are rich in electrons, and so these two atoms are able to give a chelation. They're able to chelate this metal. Also the glycine, a very simple amino acid, is able to give a chelate. But the most classical chelator agent is ethylene diamino tetracetic acid. You have used this compound in the labs when you have to determine some metals inside the solution. And we can use this compound, medicinal chemistry, this compound are used against the toxic metals in the cases of poisoning. Because you know, heavy metals normally go in the fat cells. They remain there because they are able to stay in the fat environment. And if we want to eliminate this type of toxic metals, we have to use a chelator agent as ethylene diaminotetracetic acid, because in this case we have two polar groups that can chelate the toxic metals, and these metals can be eliminated in the urine, because you see there are two polar groups that can increase the solubility in water. So these type of molecules are not so common in medicinal chemistry as a drug. Cation pi electros bond. Perhaps this type of bond is new for you. And the two counterparts, the two subjects of this bond is represented by an aromatic ring with a high electron density and a positive atom, normally a quaternary nitrogen atom, permanent charge, permanent positive charge nitrogen atom. You see in the figure the interaction between the positive charge with the negative clouds of the aromatic ring. But why this bond is so important? You see here the classical example. Acetylcholine is a permanent positive charged molecule, you see. And this molecule inside the receptor can interact with a tryptophan residue. You see, tryptophan is an aromatic amino acid with an aromatic clouds, negative charge. So in this case, you have an interaction between a positive molecule, acetylcholine, and a negative, partial negative charged clouds of tryptophan, or sometimes with a cluster of different aromatic residues. But why this bond is so important? Because normally the ionized atoms, ionized compounds, are water soluble. While the aromatic residues are lipophilic. So this bond is able to meet together two different environments. The aqueous environment and the negative charge clouds lipophilic, which represents a cluster, you know, a cluster isolated from the aqueous environment. So really, this bond is common for a drug, for neurotransmitters, because there are important evidences that a positive charge interact with the negative clouds in the target, present in the target. So this bond is called cation. You see pi on the pi electrons of the aromatic ring. So here you have a summary of the different chemical bonds, type, the energy, example, importance, you see, and other information. You see the most important, van der Waals, ionic, reinforced ionic with hydrogen bond, hydrophobic, and so cation, charge transfer, and hydrogen bonds. Okay. Have you any questions about the bonds here, atoms? Nothing? The last point, we can start with the last point of today. Perhaps then we can discuss again next Monday. The question is, how can we measure the potency of an agonist or the potency of an agonist? We have seen that we can change the chemical structure of ligands to increase their potency, because we need always of more potent drugs. But how we can measure their potency, their efficacy? This is the point. We have seen the bonds, we have seen the different approach, okay, everything. But we need a number. A number which reports the potency of a drug, the ability to interact with the binding site and to activate the receptors. This is the question. How we can measure this potency? There are two methods, a pharmacological method and a biochemical method. Pharmacological. This means that I have to measure my compound directly on the target. So, I have to know where there is my target. Where is my receptor? For example, you know that noradrenaline is able to increase the frequency of your heart, of our heart. Okay. So this means that on the heart cells there is the adrenergic, there are the adrenergic receptors. Okay, so we can take an heart from a rat, for example. We can put this heart in a physiological condition, physiological media. This means salts, glucose, amino acids, what it needs to maintain alive this heart. We can use a particular instruments to measure the frequency. Okay. So I can put inside a small concentration of noradrenaline, and I can register a number of frequency. I can add an additional, a higher quantity of noradrenaline inside, and then I have a higher increase in frequency. So this means that I can construct a graph like this. You see in the x-axis neurotransmitter, the concentration of neurotransmitter. In the y-axis, you have a percentage of the maximum effect. This means that a low concentration, I have a low effect. I can increase the concentration of the neurotransmitter, and you see an increase in the percentage of the maximum effect. But this type of curve is not so accurate. Why? You see that this curve is like an asymptote. You see, it doesn't reach the maximum. And so we have not a specific point, an accurate point, to measure the potency. For example, I can use some analogs of noradrenaline, and perhaps if these compounds are less potent than noradrenaline, I obtain another curve in the left position, in the right position, sorry, because I need a higher concentration to have the same effects. But it's difficult to compare these different curves because I am not an accurate point. There is a manner to have an accurate point. It's very simple. To change the concentration of a neurotransmitter, the concentration of our drug, in minus log of the concentration. So from the normal concentration molar, different molar, you know, millimolar, micromolar, and so on, we change the scale, no more the direct concentration, but minus log concentration. It's just a mathematical transformation. In this way, we get a different curve, sigmoid curve. Why this curve is more accurate than the first one? Because in this case, you see, we have a sigmoid curve, and in the middle, we have the inflection point. During the course of mathematics, I think you have treated this, the study of a curve. And in this case, we have a sigmoid curve, and the inflection point describes the change in concavity of the curve. So this point is accurate. This point depends from a particular minus log concentration, and we have an important information on the y axis. We have the 50% percentage of the maximum effect. So we have the so-called AZ50, efficacy concentration 50%. This is the concentration of the drug able to activate our muscle, our heart, our receptor for 50%. It is a number because it corresponds to a specific concentration. So you can understand that if we know this point of different compounds, you can imagine other curves for other compounds with different IC50. I can compare the different IC50 values of the different compounds. So I can understand which is the most potent compound. I can understand, I can have a lot of information about the binding requests. All this information can be given only by changing the scale on x-axis, only by using minus log of concentration. So this point, this inflection point is important. I can compare this value with all different curves given by different compounds. But only by agonist compound. Because you know that only agonists are able to activate the receptor, and in this case to increase the frequency of heart. So in this case, with this method, I can measure only the potency of agonists, because I measure an effect, I measure an increase in frequency, I measure a contraction of a muscle, I measure a physiological effect, the final effect after receptor activation. Is it clear? Atom? What do you say, Atom? Okay. So next Monday, we'll start again from this point. I repeat again this method, and then we will finish this part, this chapter. So I hope to see you next Monday in the meeting with all students, and then we can discuss about the course of pharmacy. Okay, questions, comments, nothing? Okay, so I close this one, and I stop the registration. Okay.